So many thanks for joining us from the other side of the ocean, and very welcome.
Thank you, and thanks for the very kind introduction. I'm not sure why I have lost my presentation all of a sudden, but I'm sure it will come back. Okay, hold on.
Yeah, we're here.
That was a very kind introduction. One thing is not quite accurate, which I'm glad to say that I am no longer director of the Center for Neuroscience, which is something I did for many years, but have now gratefully handed off. But otherwise, yes, and I only work on vision, and nothing else that you said was really true. But never mind. Okay, so let's go with that and with this. I hope everyone can see the screen. They could a few minutes ago. I'm very happy to be interrupted, but you either have to interrupt by speaking or by putting something in a chat, because I may not see you. The business of teaching and lecturing on Slack is something we're all getting used to, and although I've done some of it, it's always a process of discovery. So we'll discover this together. In any case, let me begin. What I'm going to do today is talk about a variety of issues that have interested me for a number of years, which are basic to the question of how we perceive visual forms. Now, I should say in advance that I'm not really going to tell you a great deal about visual form perception per se, but I should maybe have called this visual brain foundations of visual form perception, because I've been interested for a long time in the sort of early and mid-level processing of visual information in the brain. And that's kind of what I'm going to be concentrating on today. Before I begin, I want to sort of make note of and acknowledge two great figures who influenced me in the way I think, both of whom died this year, and both of whom have had an impact on the things that you'll hear about in this talk today. The first is Horace Barlow, who many of you, I'm sure, are familiar with. He is famous for many discoveries and many theoretical findings that relate to vision. He's the man responsible primarily for the notion of efficient coding, which dominates a lot of the way we think about visual processing. And he also wrote this extremely influential paper. It happens in 1972, which was an important year for a variety of reasons to me, not least because it's the year I started graduate school. In the single units and sensation paper, he introduced a number of critical concepts which have had an influence on the way many of us think about the brain. The second person I want to acknowledge is perhaps less well known to those here. It's Philip Warren Anderson, who is a theoretical physicist, a Nobel Prize winner, who passed away this year also. And Anderson wrote, I mean, he did many things that I couldn't begin to understand in theoretical physics, but he wrote this remarkable paper also in 1972, which I think we should all bear in mind as we think about how to understand brain function in the context of neuroscience. We tend all to be reductionists, and neuroscience is by its nature a reductionist science. But Anderson wrote in this paper what I've put on the right. I'm not going to read it out to you, but the bottom paragraph is the key one, and it's one which I think many of us need to bear in mind, which is being reductionists and understanding reductionism as a way of breaking down brain function into its component parts does not mean that we can construct a model of brain function by understanding the component parts. We need to understand things both from the level of the components and the workings of our minds and bodies, as he says, but also from the top level, because as he explains in this paper very eloquently, at each new scale at which we consider things, phenomena emerge which are different from those that you might have expected from those that emerge at lower scales. Now, one of Horace's most eloquent papers, and one which sort of leads to an inspiration and an introduction for one of the things that he gave us, was in 1961 when he wrote this brilliant introductory sentence, simply saying that a wing would be a most mystifying structure if one did not know that birds flew. So this is Anderson's thought writ large. Having a structure does not tell you what the function is without understanding what the principles of its action are. And the person who probably took this in neuroscience to its most articulate form was David Marr. And in his book on vision, he introduces, and actually in earlier papers, he introduces this notion which has become axiomatic in computational neuroscience. I think you have to put Marr's 3 levels of investigation up whenever you give a talk that's about computational neuroscience. In Marr's representational world, you have to think about 3 things. You have to think about the computational theoretical level to try and understand what the overall goal of a computation is. You have to understand the representational and algorithmic level, in which you actually ask how the goal is actually implemented in computation. And finally, you have to think about the hardware implementation. Now, modern neuroscience is concerned a great deal with hardware, but most of what I do and what I try to think about, I think, is focused on the representational level and to some degree on the computational level. And the computational challenge that we'll be trying to take on today is how do we take visual scenes and partition them into understandable components and use those components to generate behavior. The Barlow wing analogy was given new wings, if you were, by John Krakauer and his colleagues in a paper a few years ago, which I commend to all of you, which makes this clear. It explains in this diagram down below that in terms of Marr's 3 levels of investigation, you have the computation, which is the challenge of flight. That's the problem you're trying to solve. You have the algorithm, which is the flapping of wings, which is the mechanism that you use to move air and thereby support the body that's in flight. And you have the physical implementation, which is how you actually do it, in this case, with feathers. And so this sort of gives you a sort of articulation, a view of how one should think about these different levels in the context of something that we at least think we understand pretty well. Now, there is an ancestral set of ideas here, and there's also an ancestral set of theories. And I want to take a moment just to take another nod in the direction of another historical figure. In this case, somebody who was dead in 1972, had died in 1971 in a boating accident. That's Frank Rosenblatt, who was the inventor of something called the Perceptron. The Perceptron was really the first neural network. And he wrote a very eloquent paper, which is in Psych Review in 1971, which articulates the 3 questions he was trying to solve by building what he calls a probabilistic model for information storage and organization in the brain. This attracted a lot of attention in kind of an amusing way. First of all, this is the Mark I Perceptron. It is a collection of wires, and it has adjustable components, adjustable weights, which in this case are motorized potentiometers, here shown on the right. This device was on the front page of the New York Times in 1958. It is a write-up of sort of the beginning of artificial intelligence. I always like the lead paragraph, which says the embryo of an electronic computer that it expects will be able to walk, talk, see, write, reproduce itself, and be conscious of its existence, which is always a modest set of goals to think about. So the Perceptron was a simple neural network which took inputs from a retina, in this case a bunch of sensors. It collected those on a set of association units which were hardwired, and then it had modifiable connections to a layer of output units, and the key operational piece of this was that these were threshold elements which only activated beyond a certain level of input activation. Now, this is obviously a pretty rudimentary single-layer network, and its abilities were similarly rudimentary, but it is, as many of you will recognize, the foundation of what we now know today as deep convolutional neural networks. What's curious and an interesting little sidebar in history is that the artificial intelligence community was not unanimous in its enthusiasm for this device. And Marvin Minsky and Seymour Papert, 2 of the great figures in artificial intelligence, wrote a book in 1989 about, which actually had an earlier instantiation, about perceptrons, which basically argued, correctly but narrowly that perceptrons of this form could not solve a lot of interesting problems that artificial intelligence had to solve, and that this method was therefore basically unsuited to solving the kinds of problems that AI was important to solve. Now, it turns out that what they said was true of single-layer networks, not true of multiple-layer networks. And in the world that we now live in, of course, multiple-layer networks are everywhere, including not only in our computers and in our models, but on our wrists and in our phones and everywhere else. This is something we'll return to at the end of the talk to ask how much it actually has to do with the things I've been talking about. So we now have, now we should do some neuroscience, and so let's take a look at the territory in which the work I'm going to describe is done. It's the macaque monkey's visual pathway. On the left is a sort of diagram, first of all, of the pathway as it exists in vivo, that is, folded up in the brain. And it's thoughtfully been colored in here so that you can see where different visual areas of the brain live. This is a map based on David Van Essen's well-known flattened map of the cerebral cortex of the monkey. Here are the retinas, here is the thalamus, here is V1, the primary visual cortex. And this representation, which is akin to taking the visual cortex, the entire cerebral cortex, and unfolding it onto a flattened sheet, shows you where the different visual areas are. This is a couple of decades old. It's still roughly right. And it shows that the areas in color are known to have visual functions. About half of those, half of the areas and half of the surface area of the macaque brain is devoted to vision. These areas are richly interconnected, and on the bottom right here you have a simplified diagram showing the connectional architecture and, roughly speaking, the number of neurons in each of these different areas. We're going to start at the bottom, curiously, V1 and V2 here, which are the two early visual areas, and we'll spend a little time migrating towards these areas down here to the southeast, which constitute what's called the ventral visual pathway. Up here is another set of areas to the northeast, which are the dorsal visual stream. I suspect you may hear about some of this from Mel Goodale a few weeks from now in this series. But these two divisions of the visual pathway are thought to have visual functions. Form vision is primarily thought to be associated with the actions of the ventral stream. One of the interesting things to make note about here, and I think it's going to come back to be of importance later, is that although the hierarchy begins in the retina, which has actually a relatively small number of cells and fibers constituting the pathways from the retina to visual cortex, the two largest areas of the cerebral cortex devoted to vision are the first two, V1 and V2. Between them, they actually contain about half the cells in the cortical visual pathway. And that means that in a structural sense, the cortical visual pathway is like an inverted pyramid. It starts out with a very broad representation involving many neurons, and ends up with a rather narrower one with a smaller number of neurons. So areas like IT, the infratemporal cortex, thought to represent the sort of much of the final stage of this pathway, contains far fewer neurons than V1 or V2 do. And so that immediately rules out a number of ideas about how you might go about constructing complex representations in the brain, because if you were going to do that in a combinatorial sense, you would need far more neurons at the output than you have at the input. There are two other facts that are worth noting here. First, Jim DiCarlo has shown that the sparseness, that is the degree of activation of neurons in different levels of the pathway, is roughly similar. So from V1 to up to IT, if you look at the pathway while it's looking at a set of natural images, roughly the same fraction of neurons are active and roughly to the same degree. And the second thing, and this is an interesting another sidebar, is that Horace Barlow speculated quite interestingly in a paper he wrote in 1980 about why the pathway had this inverted structure, why it had so many neurons at the bottom and not at the top. Now, the question, of course, for today is how we've been trying to understand the function of this pathway. And for most of my career, I've been using what's really a kind of bizarre technique, which is the technique of recording single neurons with individual microelectrodes placed blindly into these structures, largely without any kind of functional guidance. And what we've done, of course, is to map out what we think of as the visual response fields, and we've tried to infer the function of each of these areas by measuring the visual responses of neurons. So you're all familiar, I'm sure, with the fact that in the retina, retinal ganglion cells, for the most part, have circularly symmetric receptive fields, which are sensitive to contrast and local structure, but not much else. And when we extend this analysis to primary visual cortex, as Hubel and Wiesel did starting in the 60s, what we discover is that the receptive fields of cells have developed a structure which gives them a selectivity for orientation, for spatial frequency, and for location and size, and then other factors which we won't be talking about today. So this kind of level of understanding offered to us by Hubel and Wiesel in the 60s seemed like a good idea, but then it's worth remembering that around the same time, in a sort of a very adventurous move, the next area of the visual pathway which got carefully studied was actually the inferotemporal cortex, which was studied by Charlie Gross and his colleagues, also in the 60s. And what they discovered in their experiments was something quite striking and utterly different from what we had found or what others had found early in the pathway. They found that individual cells in IT were selectively responsive to particular objects, not usually one object, but a family of apparently unrelated objects. It was very difficult to understand how these object-sensitive neurons could derive from the neurons that we knew to exist early in the pathway. And in fact, that problem of how we can take the intermediate levels of the visual pathway and convert them into the sorts of representations that are known to exist in areas like IT remains a problem. And that's the large part of the topic I'm going to talk about in the rest of the talk. Now, something else that I should say up front is that although this pathway is richly interconnected with both feed-forward and feedback projections, and that connectional diagram I showed you a couple of slides back represents both feed-forward and feedback connections, I am going to view the pathway today very largely in a feed-forward light. And there are reasons for that, and the reasons have mostly to do with the architecture of the system, which shows the connectional pattern of different areas, and also some psychophysical and psychological work, which indicates that there is very much a sort of hierarchical feed-forward structure. And this work is largely due to that of Simon Thorpe. Before I get to Simon, though, let me just show you that the visual latencies of neurons at different levels of the visual pathway, here illustrated in a review paper from Jean Boulier, increase basically level by level as we go down the pathway. Each level of the pathway is roughly represented by a cluster of areas here. And if you view this in a very fuzzy way and take a very kind of 30,000-foot view, what you see is about 10 milliseconds of delay for each area down the pathway. The vertical bars in the middle of each of these is the average visual latency for a particular study. The range is indicated by the horizontal bar. Some areas like V1 are represented by multiple studies. There are many things about this that are imperfect. The comparisons are made across different stimulus conditions and all sorts of things. But the overall pattern of increasing delay with increasing depth in the pathway is clear. Like physical evidence comes from work from, as I said, from Thorpe and his colleagues. They did a set of experiments on rapid visual categorization starting in the 90s. The subject would view a sequence of stimuli beginning with a stimulus and a mask to limit the time available for processing. Examples of the stimuli that were used are shown here. The task is always to ask whether there is an animal present in the image or not. So what you can see is there are different images at the top containing animals, here images containing natural distractors, here images containing artificial distractors. It's quite clear that there's no sort of consistent, simple feature that allows you to make the distinction between the animal and non-animal images. The task was simply to choose in a rapid reaction time design and to indicate whether the image contained contained an animate object or not. These are data from monkeys trained to do this task. Monkeys do it as well as people, but a little bit faster. The two histograms superimposed here show the reaction times for correct and incorrect judgments. What you can see is that judgments shorter than about 150 milliseconds are basically random in their correctness. But beyond about 160 milliseconds and clearly by 200 milliseconds, the monkeys are reliably choosing the animate image correctly over the inanimate image. And that means that the pathway involved in making a visual categorization decision has to be able to solve the problem in a time substantially less than 200 milliseconds, because remember, you also have to program and execute a movement in this time, the reaction time. Is a cartoon from one of Simon's papers showing the sort of idea behind this, which is that based on the latency measurements I showed you, signals pass through the pathway, gaining an extra 10 milliseconds each time. And he has this notion that the categorization relies on high-level object descriptions, and cells in IT are not responding until about 100 milliseconds after the stimulus onset. And then there's, of course, the decision and motor execution stream of the pathway. The reason that this indicates a hierarchical organization, of course, is that there is not time for extensive recurrence to go back and forth between these areas, because if there was extensive recurrence, the delays would be longer. Now, I will be the first to admit that this is not the only mode of vision. This is not the only way the pathway works, and that recurrent pathways certainly have important functions in other aspects and other kinds of vision. But this means that if you're going to take a first shot at trying to understand the visual pathway, I think it's fair to do it by taking a feed-forward view. So what's our overall problem today? Our overall problem is that we can't directly compute scenes from images. So we have a bunch of different scenes. Each of these contains multiple images on which we base perception of objects, from which we base our actions. We can't actually do this in one step. So the solution is to do it in smaller steps. And the question we ask is what representations and what computations? Now, to do this, it's helpful to make a distinction that Ted Adelson made in a paper almost 20 years ago now about the kinds of things that one finds in the visual world. And to illustrate that, I'll take a cafe in Brooklyn. I hope it's still open. I suspect it's not. It was one of Jeremy Freeman's favorite haunts when he was a student here. This is a scene, a typical scene of man-made objects as it happens. And it contains many elements which we might think of as things. It contains edges, corners of tables, the lid of a cup, and so on and so forth. And much of the way that we've been taught to think about visual processing is to think about it in terms of things, the boundaries of objects, the elements of objects, and that's kind of the way that Hubel and Wiesel and others cast their original observations that cells in primary visual cortex would be responding to the edges of objects, thereby defining the nature of things. But most of this scene is actually filled with something else, which Ted called stuff, and that is to say it's filled with things that we think of as textures, elementary texture features that are self-similar over some region of space. They can often consist of aggregates of things, but they are, in fact, the stuff of which images are largely made, and they define many aspects of the image that are important, including the borders of objects. So, for example, the change from this texture to this one defines the boundary between this curtain and the wall behind. And so we have a great deal of visual work to do in distinguishing both things and stuff and processing them together. So how should we think about this in terms of the underlying electrophysiology that we know from historical work? So Hubel and Wiesel, of course, defined the way we think about electrophysiological representations by showing orientation selective responses in primary visual cortex. They demonstrated, as shown on the left here, that the spiking responses of individual cortical cells are dominated by the feature of stimulus orientation, local stimulus orientation within the receptive field. They proposed a mechanism for this. I'll call it spatial filtering. They called it the construction of spatially organized receptive fields. So each of these diagrams here represents a map they made of the shape and size of a receptive field of a simple cell, or in this case of LGN cells, in the cat's visual system, here in primary visual cortex. But what they demonstrated was that these spatial filters would be optimally activated by targets of a particular orientation and size and position on the retina, and that would give cells the property that's shown on the left. They even proposed a circuit mechanism, which is down here. They proposed that the non-orientation selective cells of the LGN would be connected together in this particular pattern to give rise to orientation selective responses in cortical cells. You can argue, Nicole Rost has argued, that this actually represents the 3 MAR levels all in one slide. This is the problem being solved, encoding orientation. This is the algorithm, spatial filtering, and this is the implementation, the circuit detail. Now, it's also worth noting that if you go back to Rosenblatt, the perceptron, his first perceptron, was very much like a simple cell. And in 1964, he actually took considerable encouragement from the fact, as he says here, that the line and boundary detectors that have been found in the first stages of the mammalian visual system may correspond to his perceptrons, except in natural form. Now, this is a point where it's worth making the observation that there's good reason to think that this basic orientation selectivity is established by feedforward. These are data from for Jose Manuel Alonso, Marty Usry, and Clay Reed, showing that the geniculate afferents to cortical cells in cat typically have a distribution in space that's as you would expect if they were building receptive fields in the way that Hubel and Wiesel proposed. The analysis was based on cross-correlation, which in the interest of time I'm not going to go through in detail, but this shows that the thalamic inputs to cortical cells are of the kind that you would expect from a feed-forward model. And so this is, the first of cases that I'll talk about, which are in the feed-forward flavor. Now, the way we tend to think about cortical function now is more based on the idea of orientation and spatial frequency representation. Size and orientation have been sort of captured by this. This is a space representing the orientation and spatial frequency of, in this case, Gabor targets. Radial coordinates represent spatial frequency. Angular coordinates represent orientation. And just as you can map receptive fields as Hubel and Wiesel did in space, you can also map receptive fields in this space of orientation and frequency. Russ Develoy and his colleagues did that. These are receptive field maps in which the dimensions are defined by the range of orientations and the range of spatial frequencies to which individual cortical cells in monkey will respond. And it turns out that there is a direct sort of representational match between these spatial frequency and orientation tuned receptive fields and the spatial receptive fields that Hubel and Wiesel described. Here we have an idealized version of one of these receptive fields. And if we use a Fourier transform to compute the spatial filter that would give rise to this orientation and spatial frequency tuning, it looks like this. There is it's no accident that this structure here looks a great deal like the Hubel and Wiesel receptive fields that were described in the 1960s. I'll also make the observation that these receptive fields were originally conceived of as line and edge detectors, which is basically a thingish way to think about things. This notion of a texture is captured by these gratings, which are inherently stuffish. They're extended, self-similar things extending across space. And therefore, what this shows is that at the level of V1, we might expect thingish and stuffish representations to happily coexist because, of course, the receptive fields of these cells are signaling neither of those things. They're just signaling local orientation and spatial frequency content, whatever creates it. Now, we can capture all of this in a very simplified way by saying that V1 gathers information from the retinal image through the mediation of thalamic inputs having on and off non-oriented structure. There's a linear operator, a spatial filter, which is passed through a network whose purpose I will not describe, but it's needed to account for many of the phenomena of V1 responses. And then this creates a basic, simple representation of the output of V1. And we're just going to think about the output of V1 in an oversimplified way as representing orientation and spatial frequency in local regions of space. So moving on, however, the question arises of what do we know about areas downstream, and in particular, what do we know about some of the downstream areas in the pathway, V2 being the obvious next candidate. I already showed you that V2 is a large visual area. Most of it is hidden in this surface view, so you can't see how big it is. But the question of what we know about V2 is a somewhat vexed one, or has been a somewhat vexed one, because we know at the same time a great deal and not enough. So the first thing we know about V2 is that although it has visual receptive fields that are mapped topographically across the retina as V1 receptive fields are, if we look at the visual receptive field size of cells in V2 and V1 and also a few other areas as a function of their retinal eccentricity, what we discover is that cells in V2 have receptive fields that are about twice as large at any given eccentricity as cells in V1. This is kind of cartooned down here at the bottom. As we go downstream in the pathway, it's well known that visual receptive fields enlarge. But knowing that the receptive fields of cells in V2 are larger doesn't really tell us why or what they do. The notion of integrating information across space is intrinsic to many ideas about how we might process images, and it's not clear that this tells us about which particular one is done. As with much else in the visual process, Hubel and Wiesel started this whole business. They studied V1 and V2 in the cat, as it happens, and they came up with the idea that cells in V1 and V2 would be sensitive, that cells in V2 would be sensitive to slightly more elaborate features of images like the corners of objects here, shown to activate this particular example cell very well. They made little circuit models indicating how you could combine outputs from cells in outputs of V1 cells to create V2 cells that had some interesting properties. But in the years following, this particular idea has had only partial ability to explain what's going on in V2. One of the problems is that as far as neurophysiologists are concerned, cells in V1 and V2 have rather similar properties when you test them in all simple ways. So for example, these are data from two example neurons, one from V1, one from V2, showing their responses as a function of stimulus orientation, stimulus spatial frequency, and stimulus patch size, the area of the patch of grading used to drive them. What you can see is that cells in both areas, at least by this example, are tuned for orientation, tuned for spatial frequency, and somewhat tuned for size. The other way to consider this problem is one that David Van Essen and his colleagues took in studies in the early years of this century to ask how cells in V1 and V2 responded to more complex stimuli. If Hubel and Wiesel's conjecture had been correct, you might expect cells in downstream areas to be more sensitive to complex figures. And so this histogram shows, for a population of cells recorded in V2, the fraction of cells that were most selective for stimuli in one of the object categories indicated by this sort of set of cartoons at the top. And although many cells in V2 were optimally tuned for either, let's say, oriented bars or oriented gratings, there were many cells that seemed to be more interested in more complex features like chevrons, curves, so on and so forth. The problem is that this analysis does not reveal a hierarchical process particularly well. If you look at V4, what you find is, this is of course the main downstream area from V2, that you see a rather similar profile of responses. But what's more troubling is that if you go back and look at V1 using the same techniques, you see a rather similar distribution. And although I'm not going to go into the details of why I think this, I think it's arguable that the results of this experiment have more to do with the way it was done necessarily than about hierarchical transformations of information from area to area. This doesn't seem to give a lot of insight into what's going on from V1 to V2 and from V2 to V4. So we're sort of faced with this problem of we have a sort of cartoon model of V1, we have this notion of cortical hierarchy, and so the idea perhaps is to try and make a similar model for V2, but what we're trying to do is basically read tea leaves. The science of tasseography, for those who care, is the science of reading tea leaves, and what we're trying to do is really work out what this operator might be in a hierarchical model of V2. And for this, what we did was take advantage of something which I had largely ignored for much of my career, but which, under the influence of my colleague Eero Simoncelli and others, I began to pay attention to, which was to ask the question of whether V2 cells might be specialized to pick up particular features of natural images. So you can ask in a principled way what it is about natural images that makes them special, what makes them different from images you might create by drawing random numbers from a family of drawing random numbers and painting them on a screen as image pixels. The answer is, first of all, there's a well-known feature of natural images, which is that they are predominantly composed of relatively larger objects, relatively lower spatial frequencies. And so if you look at their spectral composition in terms of spatial frequency, low spatial frequencies predominate over high spatial frequencies. That, however, is not the only thing of importance. And what Eero and his postdoc Javier Portilla took note of is that when you look at a natural image, it has extended structure, which means that if you make measurements with things that might look as these do, like Hubel and Wiesel receptive fields, if you make measurements with receptive fields of the same kind in multiple parts of the same image, you are likely to be able to predict from one measurement what is going on in other parts of the image. In a random image, of course, the activation of this, let's say, particular sensor would not necessarily be correlated in any way across the image. But in natural images, there is this extended correlation structure. So images here, so sensors here be activated by this extended horizontal contour. Sensors like these would be activated by the textures and the texture gradients represented by the water. And it is generally the case that if you take any two families of textures, let's say, sorry, any two families of sensors, like these guys, and you put them in a convolutional sense across the whole image, and then ask how strongly activated they each are at the same location, you get a strong correlation between them. So this plot shows in a sort of a histogram of amplitudes the magnitudes of responses of this sensor family and the magnitude of responses of this sensor family, just by example, across a large family of natural images. And what it shows you is that when this sensor is more activated, this one is also likely to be more activated. This is not generally true for artificial images. It is true for natural and what I'm going to call naturalistic images. So what do I mean by that? So in a natural image, like a patch of a natural image here, which has a texture-like characteristic, what that means is that sensors across this image will have a correlated structure. And what Eero Simoncelli and his postdoc Javier Portilla did in work about 20 years ago was they devised texture synthesis methods which took account of not only the overall spectral composition of this image, but also these correlated features. So on the left, you have a texture created by synthesis from this image, which was done by taking the same orientations and spatial frequencies and basically jumbling their phases to create an image which has the same spectral structure. We can call it spectral noise or just noise. And on the right is a naturalistic texture, also generated by synthesis from the same image, which has the same higher order correlations as well as the same spectral structure. And if you do this with families of natural images, on the top row, what you get are synthetic images on the bottom row, which are designed to capture that same spectral structure. As you can see, for things that are texture-like, hair, lizard skin, rocks, pieces of stone, the synthetic images have a good resemblance to the natural images from which they are derived. This doesn't work for faces. Richard Feynman here doesn't do very well when he's texturized. But notice that what's happening here is that every local patch of this image has a Feynman-like texture. It's just that the global structure has been disrupted. And what this means is that this texture synthesis method is not appropriate for copying images. It is a method for copying image statistics. And this is where we're going to be thinking about how to make this into a principle of organization. Now remember this. Remember that V1, V2, V4 all have different characteristic receptive field sizes, and this actually turned out to be the feature of these images that that led us to look specifically at V2. So this is a demonstration that Jeremy Freeman made based on a photograph of people in Washington Square Park. I'm going to ask you to fixate on the red dot, and what I'm going to do is I'm going to set an exchange going. I hope this renders properly in the world of Zoom. You're seeing two images exchange, and if you fixate here, I think you won't be able to distinguish those two images from one another. But if I then draw your attention over to this part of the image, what you'll see is that in the two images, one image has all of its local patches of image replaced by matched textures created by the same algorithm. And what Jeremy and Eero did was they explored the size of this texture patch, which could be manipulated without human observers noticing it. And what they discovered was that it was the V2 receptive field characteristic, which defined the size of the patch, which was just barely detectable. And that's what suggested that V2 was the area in which one should look for these effects. So for experiments in this and all later experiments, what we did is we took a bunch of natural images here, and we used the texture synthesis method that Javier and Eero had devised to create textures which were either naturalistic textures, which contained all of the oriented structure and the texture statistics, or noise textures, which were the same textures, but in this case, only the spectral characteristics were matched. And then we'd string them together into a movie like this, which is a series of the 15 different textures, 15 different families naturalistic and noise all run together. And then what we would do is measure the responses of cells. And our basic original data set was to measure the responses of cells in V1 and V2 to these textures. These are the two example neurons whose data I showed you before. These are the responses that they gave to naturalistic in solid and noise in pale colors here. And what you can see is that the naturalistic texture evoked a much stronger response in the V2 cell than the noise texture, not so in the V1. Across the population, that is also true. What you have here is a time mark for where the texture is presented. These are the average responses of about 100 V1 cells and 100 V2 cells to a whole bunch of different families of texture, divided up in each case by naturalistic and noise. The naturalistic textures evoked substantially stronger responses than the noise textures in V2, not in V1. And this plot over on the right shows the distribution of what I'll call a modulation index, just simply the difference between the naturalistic and noise textures divided by their sum, showing that V2 had a strong modulation on average. About 2/3 of the cells were significantly modulated. V1, not so much. Now, I'm going to, in the interest of time, pass over one piece here, which shows simply that there is a relationship between this feature selectivity and some of the compartments of V2. I'll be happy to come back and talk to people later about that if you're interested. But I'm going to consider next whether there's evidence for a feed-forward circuit architecture in this process. So this is a sort of canonical diagram of a piece of cerebral cortex showing the different layers. The standard architectural picture that we all work with is that thalamic inputs come into layer 4 and are processed by supragranular and infragranular circuits and then passed out to other cortical areas. It's the supragranular layers that we think of as providing the main output to other cortical areas. In our original study, we had found that cells throughout the layers of cortex of V1 did not show this texture modulation, but cells in V2 did. But so what we decided to try and do was ask whether we could learn something about the dynamics of this response by making recordings with these multi-electrode arrays. This is a 32 contact multi-electrode array, which is placed in this diagram so that it crosses V1 and V2, which you can do conveniently because the macaque monkey's cortex is laid out the way it is, with a single electrode penetration coming in from the surface of the brain. What's shown here are firing rate plots for multi-unit activity recorded from this electrode as it passes through V1, white matter, and through V2. In each case, the texture response is shown in red. The spectrally matched noise response is shown in black. And over on the right here, we have a D prime measure of the difference between these two traces. And the short answer to look at this just briefly is that in V1, there's rather little difference between the responses to the two textures. And in V2, there's a robust difference between them. And you can see that either by the big red blotch here or the big difference between the red and black traces over here. Now, this is one example track. If you take data from multiple penetrations and shown here, you can also see the same kind of effect, but you also see some interesting dynamic features which are suggestive. In each case here, the black and the red traces are as they were before, the responses to naturalistic and noise stimuli. The green trace here for V1 and the blue trace here for V2 is the trace showing the evolution of sensitivity to the difference between these two texture families. And you can see that in V1 there is some sensitivity, but it is delayed and comes up late in time. In V2, the texture sensitivity emerges very rapidly, almost as soon as the visual responses begin. And if I just pull these blue and green traces out to the side and superimpose them, what you can see is that, in fact, in all cases, the response in V2 to the difference between the two textures precedes the response to V1, here by 20 or so milliseconds, in the upper and lower layers by a somewhat smaller amount. This dynamic structure suggests to us that the way the computation is being done is that signals arrive in V1 through layer 4. They are processed, as we know, by supragranular and infragranular circuits. There is a projection from both of those circuits to layer 4 of V2, and then there are, of course, recurrent computations within V2. This dynamical structure suggests that the first place that naturalistic texture signals emerge is in layer 4 of V2, as you would predict from a feed-forward model.
Can I ask a question about the latencies? I mean, I see here that the V2 latent. It seems that they are in a way preceding, at least in some way, V1. And I'm wondering, in the earlier slides that you've shown with earlier studies looking into V2 latencies, they do seem to have longer latencies than V1, but do you think it might be because they were not targeted with appropriate stimuli?
No, so it's a good question, but it's because I went too quickly. Remember, this is the latency of the response to the difference between the two textures. If you go back here, what you can see is the actual onset of the visual response in V1 is at about 45 or 50 milliseconds, and in V2, it's about 10 milliseconds later.
Oh, okay, so it's the delta. Okay.
So what happens is that in V2, you get the texture response right away. In V1, it's delayed.
Okay, thanks.
Sorry, I should have explained that more carefully. Okay. Now, so one question now at this stage, and I think so I've got about 12 minutes left, and Sharon, you could tell me how much leeway I have on that.
I mean, we're flexible. I mean, if people have to leave, then they will leave, but you can, it's okay. No, we're not in a conference where there's another talk after you.
I'm glad to hear that. Well, actually, I would have been interested to hear the other talk. But, okay, so I know what to do here.
Next week.
Can I also ask a question? Like, in cases where your target has differences in the structure, then it increases the delay. There is like more recurrence compared to some targets that they don't differ in the structure.
So I'm not sure I understood the question, but if basically that reflects back, I think, on the sort of Thorpe feed-forward analysis, I think the answer is that you can do quite complicated visual discriminations in one pass through this pathway. But of course, the kind of vision that requires scrutiny and sort of longer inspection presumably does involve more recurrence. Is that the kind of thing you were aiming at?
Yes. So actually, the delay changes according to the task that
yeah, we have no evidence that the delay of neural responses changes with task, at least not in the parts of the pathway I'm talking about today. We do know that the delay does emerge, of course, by the time you reach decision centers. But as far as early visual representations are concerned, I don't think there's any evidence that the task alters the delay of visual response.
Okay, thank you.
So one way to think about what's going on is to try and build a simple feed-forward model, and I'll just cartoon some modeling work based on these same ideas I've talked about, which is the idea of local derivatives. Horace Barlow introduced this idea of redundancy reduction. This is Horace, and Horace applied Horace with a local derivative applied to his image, showing you, as many of you know, I'm sure, that this enhances the borders and contours in the image and de-emphasizes regions where the image doesn't change. So the construction of visual receptive fields as a set of derivative operators is an idea that sort of goes back to Barlow and Hubel and Wiesel. In the sort of classical canonical building of a V1 receptive field, we're creating a simple spatial operator. And what you can do is think about the way that these spatial derivative operators process images is that the image is everywhere, oops, that's not supposed to happen, is that the image is processed by a series of these local derivatives everywhere. And that is what creates the cortical representation of this image. Now, in V1, we know there are two kinds of cells. There are simple cells and there are complex cells. I haven't addressed that structure at all, but complex cells seem to be driven by families of inputs from simple cells of similar properties. And if you simulate this network of simple and complex cells and then simulate its responses to differences between naturalistic and noise textures, you find that there is basically no difference. Simulated V1 cells give identical responses to naturalistic and noise textures, very much in the same way that our real V1 cells did. So we were interested in asking whether V2 could actually compute these texture properties on the basis of derivative operators. But the map that V2 sees is not the simple map of the original image that we imagine that V1 gets more or less unmodified from the LGN. What you have instead is a map based on the representation that V1 computes. Now, V1 computes a representation based on orientation and spatial frequency. Now, this picture is it's a little bit of eye candy. It comes from the work of Ed Calloway's lab, and it shows the spatial representation on the surface of the cortex. This is a span of a couple of millimeters in each case, of orientation and spatial frequency maps. So you all know that there are orientation columns, and there are regions of local spatial frequency. So imagine then that you took derivative operators and executed derivative operations not on the image of the children, but on the actual receptive field map structure of V1, so that this derivative would take, for example, the difference between structures of two different orientations and a third orientation in the middle, or structures of different spatial frequency and a third spatial frequency in the middle. The concept here is that this derivative operation can be thought of in cascade from stage to stage in the cortex. And if you do that, you can create V2 simple cells, so-called, by pooling inputs from a bunch of V1 cells, let's say preferring different orientations. That's what's going on here. And if you create a V2 simple cell by pooling across orientations and then ask whether it actually has a texture sensitivity, the answer, it does not. However, and this is the point of this simulation, if you create a V2 complex cell by pooling a bunch of self-similar simulated V2 simple cells, what you find is that these V2 complex cells have a responsiveness to the texture stimuli which is similar to the response we measured in V2. The idea here being that you can actually simulate this sort of transformation into the texture statistic sensitivities in this sort of derivative kind of model. And so what you have are cells that are in V2. These are simulated neurons, and the simulated V2 neurons actually have properties that are similar to the real measured neurons in these simple domains as well. So what that means is that with a simple cascade of derivatives, it's possible to create a synthetic image process that kind of mimics what we know V2 to do on the basis of experimental data. So let me try and wrap up in the next few minutes by asking what happens when we go beyond V2, because of course V2 is only a small way to IT. And in this there are couple of different things that I'm going to show you in brief. One is data from other labs looking at the responses of cells in V4 in light of a process that can be helpful for us here. And so these are data collected by Goki Okazawa when he was a graduate student in Komatsu's lab, looking at the responses of cells in V2 and in V4 to texture stimuli and noise stimuli created in a method similar to the Portea-Simoncelli method. What you can see here is that the response in V4 is on average larger than the response in V2, and the discriminability in V4 is also a little bit larger than V2. That suggests that this processing of texture continues in V4. But what V4 also does, which is something that we don't think V2 does, is it actually becomes sensitive to the structure of the images from which the textures are synthesized. So these are data collected by Nicole Rust when she was a postdoc in Jim DiCarlo's lab. Again, these are responses to natural images, in this case not textures but natural images, and to the scrambled versions of those natural images created by the Portea method. Down here is a plot showing the results of the experiment, which were cast in terms of the ability of neural populations to distinguish different natural images from each other or different scrambled images from each other. And in the experiment, they actually compared V4 and IT. It's easier to think about IT first. Remember that IT cells are very sensitive to actual natural images in stimuli, and they don't respond well to simple targets. So what this shows is that IT does a much better job of classifying natural images than it does of classifying scrambled images. What this also shows, though, is that V4 is also better at classifying natural images than it is at classifying scrambled images. The difference is less, and if you extrapolate back, which is highly unwise, but in this case we can imagine extrapolating back to V2, you might expect to see no difference between natural and scrambled images in the responses of V2 cells. So this suggests that in V4, you have an enhanced representation of texture information, and you also have the beginning of a sensitivity to, let's say, things rather than stuff, going back to the Adelson philosophy. There's evidence for this from work that Anne Hermanstadt and her colleagues have done, showing that if you create maps of images like this based on the boundaries between textures, remember I pointed out that texture boundaries are often an important cue to form, that you can actually define object boundaries from these derivatives of texture families and show that objects can begin to be defined on the basis of the boundary between different texture regions. So we were interested then in the question of how to ask beyond V2 what these different representations actually look like. There are a couple of problems that are worth mentioning here and a couple of points that are worth making. First of all, it's very difficult to use natural images to study individual neurons, because when you study the receptive field of an individual neuron, it lies somewhere in a natural image. You don't know where. And in order to understand how populations of neurons respond to natural images, you have to do the analysis the way that Rust and DiCarlo did it here, which is by studying the responses of receptive fields that live all over the image. Textures by nature are self-similar. Natural images by nature are not. So we never actually studied the responses to natural images in our V1 and V2 cells because we thought the technical challenge was beyond what we could do with single neuron recording. Now with populations, we can actually overcome that, and we are, of course, along as many others are, looking at population responses to natural images. But of course, the technique that's ideal for looking at populations of neurons on a large scale is functional MRI. And so what we did was we jumped from monkeys to humans and from single electrodes to the magnet to study the responses of human brain to the kinds of stimuli that I've been talking about so far. So let me remind you that our textures were created from original ancestral forms. Here's that picture of those kids. Here's a picture of a honeycomb. If we process those textures using the Portia-Simoncelli algorithm, if we just match the spatial frequency content, we get what we're calling noise here. So these are the two noise textures. They are, as I've already shown you, not very similar to the ancestral textures, but they do have the same orientation and spatial frequency content. So here's the ancestral natural image, here's the noise. And down here at the bottom, of course, is the naturalistic texture, which includes not only the same orientations and spatial frequencies, but also the correlational structure. And these naturalistic textures, as I showed you before, look a lot more like the ancestral natural images than do the noise textures. Notice that the naturalistic texture looks quite a bit like an ancestral image when it's a texture, like the honeycomb. The naturalistic texture does not look a lot like a picture of a bunch of kids standing around. So this is, again, the fact that this is not about copying images, it's about copying image statistics. So in order to do the MRI experiment, we did nothing fancy. It's a pretty simple design. We're interested in comparison of the responses of early visual areas to visual noise and naturalistic textures. And to do that, what we did is we did a straightforward MRI block design. We'd show flashing textures for 9 seconds, flashing noise images for 9 seconds. The subjects were fixating an uninteresting image in the center and doing a task to confine their attention to the center of the image. The flattened map of the posterior pole of the cortex of one observer is shown here, and the area boundaries were separately established by a topographic mapping experiment. This is the standard flat mapping technique. So if you then ask how the responses of this cortex measured by the fMRI varies according to fluctuation between noise and texture stimuli, the answer is here. What you get is rather strong modulation, association with the exchange between texture and noise in V2, V3, V4, and in areas dorsally anterior to V3 and V4, which are the lateral occipital complex of the human visual cortex. In V1, there is no modulation. So as in the neurophysiology, cells, or the MRI signal, the bold signal in V1 appears to be insensitive to the exchange between textures and noise, and our interpretation, of course, is that that's because they have the same orientation and spatial frequency content. Now, you can also compare natural images to noise. Others have done this experiment, but not in exactly this way. Again, you have the same orientation and spatial frequency content, but of course, this image contains a bunch of things, in this case, the kids. This image does not. Now what you have in this measurement, again, we're comparing images with noise, and this exaggerates the responses in downstream areas, and we'll see that in a more direct comparison in a moment. And again, V1 is still not strongly activated by the difference between these two images. That is to say, in another way, to be very, very blunt about it, V1 doesn't care at all about the difference between things and stuff as long as they have the same orientation and spatial frequency content. But the interesting comparisons, of course, are when we compare the responses of these cortical areas to naturalistic textures, which are, remember, stuffish representations of the image statistics contained in this image, and the actual original ancestral natural images, which are the images that contain the things, if you like, that makes this image an interesting part of the world. When you do that, what happens is that this exchange fails to activate V1, as you would expect, but also fails to activate V2 and V3. The first areas that show strong activation to the exchange between natural images and the texturized version of those natural images are V4 and the areas anterior to V4, the lateral occipital complex. Recall the Rust and DiCarlo observation, which showed that V4 was a little bit better at classifying images containing objects than it was at classifying images containing textures. We think that that's what's going on here. And finally, if you put all three of these things side by side, you see that the pattern is quite orderly and quite organized. Textures versus noise, that is naturalistic textures, evoke stronger responses in V2 and V3 especially, not so much in the anterior areas. Forms, the actual original images versus noise, have a similar pattern of activation in V2 and V3 and stronger activation in downstream areas. And finally, if you look at forms versus textures, what you see is effectively the difference between these two left-hand images. In the right-hand image, you get a strong activation of the areas of the lateral occipital complex, which correspond to the areas that we call the IT areas in monkeys. So to sort of cartoon this, you might imagine that V1 and V2 represent spectral statistics, textural statistics, and finally form statistics, if you will. If you want to go back to Ted's paper, V1 and V2 are stuffish, and only when you reach V4 and IT do things become thingish. And just to wrap up in the last couple of minutes, I do want to draw a parallel because I introduced Rosenblatt to you and I gave you perceptrons. And of course, the world of perceptrons has now become the world of convolutional neural networks. And it's of interest to many of us to wonder how well these networks, which are built in an architecture which was designed in many ways to be neural-like, using operations that are thought to be neural-like. This is sort of a cartoon of a generic neural network, starting with an image, and it has the task of classifying the elements of an image. It contains features like linear filters, rectified linear units, spatial pooling, downsampling. The normalization here is not actually the same normalization, this is the same term used in a different way from the way neurophysiologists use it, but these are neural-like computations. And so the question is, of course, whether the calculations in these networks are like the calculations that the brain does. Dan Yammans and Jim DiCarlo have made the argument that different layers of these multiple network models do have features that resemble different cortical areas in terms of their responses to forms. And it turns out that Dan Yammans and some of his collaborators looked at the responses of 3 of these characteristic networks to textures and showed that the first layer of these networks did not differentiate textures from did not differentiate noise textures from naturalistic textures, as V1 doesn't in our hands. Layer 2 and layers downstream of this, in all of these networks, did respond in an enhanced way to naturalistic textures compared to noise textures. This is not, in my view, a strong argument. This is a looks-like argument, but it does suggest that the elaboration of texture information is something that is useful for structures that are analyzing images to do in order to go from the representation of stuff, if you will, to the representation of things, which is the goal of these complex neural networks. So what I've tried to tell you is that we're beginning to get an understanding of some of these intervening stages in the pathway that transform information from the elementary representation captured in the retina to representations of objects. Are we there? No. Are we approaching an understanding by matching the different stages of the calculation in brains and machines? Perhaps. I'm confident about what we're learning about the brain. I'm less confident about what others are learning about machines, but that's because it's not my territory. It's clearly an interesting topic and place for discussion. And I will again just remind you that I want you to concentrate on the idea that the representation and algorithm levels of the MAR models are the kinds of things that I think are most helpful in understanding how this is done. I wouldn't want to discourage anyone from studying the hardware implementation, but what Philip Anderson's More is Different paper tells you is that it's very hard to deduce the structure of a large thing like a computational network from the structure of the hardware that it contains. And so I think representing knowledge about the brain in all three of these levels is critical. Let me just finish up by showing you the people who did a lot of this work. Jeremy Freeman and Corey Zemba were students in the lab during the early parts of these experiments. Corey is now a postdoc at UT Austin. Jeremy has, I think, is now self-employed, saving the world from the effects of climate change, but he has also worked at Genelia Farm and at the Chan Zuckerberg Institute. Javier Portilla was a postdoc with Eero Simoncelli, who did the original texture work. David Heeger collaborated with us on a bunch of the MRI experiments. Julia Pai and Richard Perez were both involved in the laminar analysis. Julia is now a grad student at WashU in St. Louis. Richard is an MD PhD student at UCSF. And Eero Simoncelli has been my close colleague and indeed inspired a lot of this work all the way through. And he is, of course, not wielding the electrodes, but he does pretty much everything else. So with that, I will stop and thank you all for your attention. Apologize for going a few minutes over. And of course, I'm happy to take any questions that didn't come up in the course of things. I think what I'll do is I will stop sharing unless a question calls for a visual.
Thank you very much, Tony. Let's all, you can unmute yourself and let's give a big applaud to a wonderful and insightful talk. Very interesting. And there was one, I'll start with one questions that appeared in the chat by Sarit Shapiro. She asked if, like the different orientation tuning in V1, if it is known or if you've investigated different types of texture tuning in V2.
Yeah, there is a the notion behind the derivative model is that individual V2 cells will combine information from different kinds of components in V1. And so we are at the moment doing an experiment. I had considered trying to put some preliminary data in, decided not to. Tim Alescu, who's a postdoc who's working with me and with Eero Simoncelli, is doing experiments recording from V2 cells in behaving monkeys and then using a derivative model to try and fit the data from each of those cells. And the principle behind that model is that you have a set of inputs, which are V1 inputs, that represent different orientations and spatial frequencies, and the V2 cells execute different nonlinear combinations of those inputs. And each V2 cell is basically free to choose, if you will, which V1 inputs it combines. So the idea here, at this stage it is an idea, not a proof, is that the different properties of different V2 cells will be captured by the different way in which they combine inputs from V1. And you can imagine that working in a couple of different ways. The derivative model, which I kind of blitzed through, implies that there are actually 4 dimensions represented in V1, which are combined by V2 cells. There's position, X and Y, and there's orientation and spatial frequency. So imagine how some of those different combinations might work. 2 V1 cells that are in different positions and different orientations would define a corner or curvature. 2 V1 cells that had different spatial frequency preferences, but the same orientation preference and location, would represent an edge, because an edge is, of course, multiple spatial frequencies coincident in space. And so the idea, and it is still an idea, is that different V2 cells will combine different elements out of this 4-dimensional input space from V1 and create an operator. Now, I should also just go back to the original question, which is to ask whether different V2 cells have a strong preference for different textures. The answer there is a little more complicated, because the thing that you have to bear in mind is that cells in V2 are sensitive to orientation and spatial frequency as well as spatial statistics. In fact, you can argue that most of their responses are driven by their preference for orientation, spatial frequency, position, and so on and so forth. So V2 cells respond to different textures in part because those textures contain the orientations and spatial frequencies that they like. Then on top of that preference, there is this layer of texture statistic selectivity. And what I can't say for sure is that cells in V2 that have a particular orientation and spatial frequency preference come in multiple texture flavors. It's an interesting question, but we don't have enough data to make that, to answer that. That may be more of an answer than you were hoping for, but
no, definitely.
Opens
thorough answer, great. Yeah.
Tony, I have a question for you. In your model, you're taking the feed-forward local processing. So I'm puzzled what far we need the horizontal connection in V1 if we don't completely use them.
Well, Uri, I mean, you and I have talked about this kind of thing many times. My belief is that the horizontal connections in V1 are primarily there for the construction of the classical receptive fields that we measure. That is another story. But there is, however, evidence that lateral interactions do contribute in a feedback way to the selectivity that I talked about. I actually took out, sadly, about 10 minutes of a story, which I'll try and summarize briefly because I think it does bear on this question of recurrence and lateral connections. What Korizemba did was he asked whether the size of the texture actually influenced the texture selectivity of the cells. So I think you're all aware that V1 cells have receptive fields of a particular size, and as you enlarge the stimulus beyond the receptive field, you typically see first enhancement and then suppression of the response. And what Corey discovered is that the suppression of the response is much greater for noise textures than for naturalistic textures. So to turn that upside down, what that means is that the optimal stimulus size for a naturalistic texture is large compared to the receptive field. And the implication of that is that part of that response actually derives from cells that have larger receptive fields that have recurrent connections to the cell that we're actually studying. So in other words, if you look at a stimulus that's only in the classical receptive field, it has some texture, there's some texture selectivity. This, of course, I'm talking about V2. But as you enlarge it, you actually get an enhanced texture selectivity. And we think of that as at least partly reflecting feedback from V4 to V2. It's also true, and I didn't emphasize this in the dynamics, what you saw is that in V1, there is a texture response, but it's late. That could emerge either from horizontal connections in V1 or from recurrent connections from V2. So even though I'm a confirmed feed-forwardist, there's plenty of stuff in the data I showed you today that demonstrates that a simple feed-forward, non-recurrent architecture is not enough to explain everything in the data. But I only had an hour, so let me do what I can with an hour.
Thank you.
Tony, can I ask a question?
Yes, of course.
Great talk. I'm wondering how you think about the thing selectivity in LOC that is really dramatic. Do you think that it is generated by some clever combination of your texture neurons in V2, or there are separate independent channels that are, remain selective to the lines or to the contours and are not contaminated by the textures, and they are the ones that actually drive it? How do you think about this? Because, we have a mosaic in V2 of
Right. So several good questions in that question. So the first thing is, if I were to actually wonder about that, I would probably ask you, not me. But since you're asking me, I can't do that. What the conjecture is that the same kind of calculation that converts the V1 representation into the V2 representation, which is a sort of spatial pooling and derivative across the representation, if cascaded, will automatically generate selectivity for objects by increasing the scale and the specificity of the range of connections. That is a conjecture. We haven't demonstrated that. It's an idea we're trying to explore, planning to explore in V4. If it were not for COVID, we might have actually started, but at the moment, it's a conjecture. However, I don't think there's any particular evidence that is correct. what you learn, I think, for me, the main lesson about convolutional neural networks is you learn that repeating the same simple operation over and over again can create surprisingly complicated representations. So the intuition that we're working with is that the same kind of cascade of operations that we know works at some level in convolutional networks also works in brain networks. Now, I do want to respond, though, just finally to one thing that you said, which was the idea that there would be responses, you said, I think, uncontaminated by texture in the early visual pathways. I don't think there are any responses in V1 that are uncontaminated by texture, just because I don't think V1 cares about the difference between contours and textures.
I was talking about V2, not V1. But your natural textures.
Okay, fair enough. Yes, so there are cells in V2 that don't care about naturalistic textures. Yes. And of course they do exist. We don't know why they're there. We think they're actually in the thin stripes of V2 in the compartments. We have one of the we have some evidence from optical imaging work that Corey has been doing in Texas with Ian Nauhaus and Robogoras, that they are confined to two of the three stripe compartments. Yeah, so circling back to your question, the answer is yes, I believe and hope that cascaded operations of the kind that we have already seen V1 to V2 will work, but I can't prove it to you.
Okay, great, thanks.
Tony, there's a question. Daphne Moore has asked about if you have looked into what happens in V3, or I would say VP, because it was like you went from V2 to V4, and I was wondering if you have any information about VP, or she's wondering actually.
It's a very good question, and it's a question that all people who work on human brain ask of all people who work on monkey brain. And here's the thing, okay? The answer is extremely simple and very dull. V3 in the monkey brain is entirely buried within a big sulcus called the lunate sulcus. It's hard to find, and it's rarely studied in detail. What's happened is that the whole visual form processing community jumped from V2 to V4 because they are exposed on the lateral surface of the brain, and they are relatively easy to find. It's a lousy excuse. It's a very good question. What the human MRI data suggests is that V3 looks kind of like V2 in its responses, but that's obviously a very sort of crude overview. Mm-hmm. We do know some things about V3 which suggest that it has a strong role, at least in monkeys, in visual motion processing. That is, it has a stronger association perhaps with the dorsal visual stream. But Daphne, your question is well taken, and I can't really answer it effectively. I guess the answer is I hope it doesn't do anything really interesting that V4 doesn't capture.
Okay, thanks. Udi.
Yeah. Tony, I was wondering if you can, if your models explain also our understanding of the gist of the scene. For instance, if we know if the scene's indoors or outdoors, and because you never referred to this, you were talking about object recognition specifically, and this is sort of because this happens very quickly, and maybe also would happen, it happens within what 100 milliseconds or less than that. So I wonder what you have to say about that.
So let me, Unfair, let me ask you a little bit. What exactly do you mean by you're talking about the gist of the scene?
I'm saying the ability to discriminate between, is it an indoor scene or an outdoor scene? The ability to parse the scene into different elements in the scene, say the sky, the background, and so on and so forth.
Yeah, so it's a great question, and I don't think I can answer it, but I can tell you that I think so. It's akin to the question that Simon Thorpe is asking. I think he chose scenes containing animals and scenes not containing animals as a sort of shorthand for the gist of a particular kind of scene. And that experiment, although I passed through it as though it were an accepted set of truth that everybody buys, has actually been quite controversial for a reason that I'm sure you know well, which is there a set of low-level cues in images containing animals that distinguishes them from images that don't contain animals? And there's, Felix Wichmann and others have done careful analyses of the image sets that Thorpe uses. There's sort of, I don't know if it ever resolved, but there's kind of a rumbling discussion about that, which I'm aware of. The scene gist question, I think, is similar, because there are many things that the visual system can do effortlessly on the basis of low-level cues. So if you ask me whether it's a night scene or a day scene or an indoor scene or an outdoor scene, I might be able to do that on the basis of very simple statistics like things that V1 could encode. And one of the things that the kinds of ideas that I've offered you today discounts is the fact that, as you know and as we all know very well, all areas of the visual cortex have outputs to the motor system. V1 projects to the motor system. V2 projects to the motor system. All of these areas have separate pathways out. So we imagine that elaborate processing of form requires that information percolate down the pathway to somewhere like IT and then leave. But if there's a simple cue that could be encoded, let's say, by V2, then there's no reason to believe that the information has to pass all the way down the pathway in order to create a behavioral response. That kind of gets close to the question of where visual awareness resides, and that's another whole thing that I think is beyond my, sort of above my pay grade. But it highlights the sort of the limitations of a purely hierarchical way of thinking about the visual pathway. So question well taken, bad answer, but it's the best I can do.
Can I ask a question? So I wonder what you mean when you say that V1 doesn't care about contours. It means probably in terms of response activity, but maybe there is some different coding, maybe temporal coding of the grouping of contours, some other way of representation that you don't measure.
Possible. I've not found compelling evidence for that kind of coding in issues like contour grouping. So, for example, there are reports, and many reports, of things like synchrony arising in V1 populations associated with different sets of image statistics. People may debate how important those timing codes are, but there seems to be something there. Now, what I think no one has demonstrated is that those codes are unique to contours. I'm pretty sure that if you had an interaction between two V1 receptive fields and you stimulated them with two parts of a line, or you simulated them with two pieces of texture that matched, you would probably get the same kinds of synchrony. At least I have no reason to think that they would be processed in a different way. So, or is there evidence that V1 does things cares about things over space? Yes. Is there evidence that it cares specifically about contours over space? I think that's harder to say. I think that there are. I think it's genuinely the case that the things that V1 does don't care about the difference between objects and textures, just because of the way that the elementary receptive fields work. It could be proven, but the experiment you would need to do is one that no one's done. So I'm sure you remember, Singer back in the day did these experiments with broken separated lines as compared to continuous lines, basically showing that if you activated a whole swath of V1 at the same time, you could get these synchronous responses. I'm sure if he did that experiment with gratings or with moving textures, he'd get similar results. And so that would suggest it's not the line that matters, it's the continuity of the region of activation. Anyway, that's my story and I'm sticking to it.
Okay, so let's, are there any more questions? Or in any case, let's again thank you, Tony. It was a great pleasure to have you with us. One big benefit of the COVID is having amazing speakers like yourself. And
well, thank you all.
Thank you very much. It was a pleasure.
This has been probably one of the most distinguished audiences I've ever had, even though I can't actually see any of you in person. So it is a compensation of sorts. It's great to see you.
Maybe it's good time to say, to ask all our distinguished guests to distribute this list about this vision seminar, because it became so simple to be global and international. So we are very happy to distribute it.
Yeah, it's good. You're part of this worldwide neuro thing, which actually does distribute that information pretty widely. So I think a lot of the community is kind of keyed into that list and mechanism.
Yeah, definitely. Amazing initiative. Thank you very much, Tony.
Thank you. Thanks for the invitation. I'm sorry I couldn't visit.
All right.
Okay.